DNA profiling in rape cases in Malaysia. Introduction. Since the 1980s, one of the scientific discoveries that have had a particularly notable impact on criminal investigations is the use of highly varied DNA sequence sections, or known as DNA profiling, in the human genome for individual identification, which was formerly known as DNA fingerprinting. DNA profiling is defined as the process of obtaining a DNA pattern known as a profile from a sample. The DNA profiles of the individuals can be used to identify and compare them. Some of the significant use of the DNA profiling are to determine the likely origin of a bodily fluid sample linked to a crime or crime scene using DNA profiling, to shed light on family ties, and to locate those who have been affected by a natural calamity. DNA profiling for criminal cases is handled lawfully by the DNA Identification Act of 2009 and the DNA Identification Regulations Act of 2012 in Malaysia. Only qualified professionals appointed by the Minister of Home Affairs can examine DNA for a legal case, and their work must take place at Royal Malaysian Police and Kimia facilities. Now, let's move to the part where techniques used in DNA analysis are introduced. A few of the most common biological evidence acquired in rape cases are semen, vaginal fluid, blood, hair, sweat, epithelial cells, saliva, and other body fluids. Biological evidence can be crucial in establishing the presence of sexual contact, elimination, or conviction of suspects by comparing the acquired evidence with an example. DNA analysis remains the most common identification method for biological evidence. Here are two techniques used in DNA analysis discussed. The first one, polymerase chain reaction, or known as PCR. PCR generates millions of copies of specific sequence of DNA by using only a small amount of it in a short of period. It is also useful in forensic science, especially if it involves rape cases where the amount of DNA is limited. It also uses cells process for replicating a new DNA strand. Second is autosomal short tandem repeats, or known as STR. STR evaluates specific regions on the DNA specimen known as losses. It can also provide evidence of a person's identity. It also has a higher level of variance. Identification of perpetrators, thus providing solid evidence for legal matters. Issues and challenges. There are a few issues and challenges occurred. The first one, insufficient data for DNA profiling in Malaysia. Second, contamination. Third, unreliable techniques. Fourth, wrong handling of evidence. And lastly, DNA evidence planting and fabrication. Insufficient data for DNA profiling in Malaysia. Forensic DNA Data Bank of Malaysia, or known as FDDM, is a database that stores DNA profiles that can be used in investigations by comparing the DNA profiles electronically. The data bank can be useful in linking or identifying suspects to the DNA evidence collected at the crime scene or from the victim. It can also be used to exclude suspects as well as link crimes to each other by the shared DNA profiles found common at the crime scenes. Moreover, the DNA data bank can be a tool in not only solving crimes but in crime prevention by monitoring repeated offenders and avoiding false accusations as well. The implementation of FDDM by the DNA Data Bank Division of the Royal Malaysia Police and the Department of Chemistry Malaysia is a success, as a total number of 160,534 DNA profiles were obtained and stored by 2019, which is a huge rise from 2017, where 70,570 DNA profiles were stored. 69 hits were obtained by using FDDM in 2019 for violent crimes, and this shows a surge compared to 2017, where only 17 matching cases were found. This contributed to the highest number of hits. The notable here thing that only around 30% of convicted individuals' DNA profile is stored in FDDM. However, there are also a few downsides. First, database on race. FDDM lacks a comprehensive DNA database for Proto-Malays, Senoi and other ethnic communities in Sabah and Sarawak. The identifying male DNA evidence in vast epithelial cells or in extended postcoital intervals. Second issue and challenges is contamination. In a forensic investigation, contamination might happen in two stages. First, during the investigation of a scene, and second, in a laboratory analysis. However, contamination in crime scene evidence is not only restricted to people involved during the investigation, but also may be affected by environmental influence such as snow, rain, wind, temperature, and humidity. Contamination can also occur in laboratory. For example, cross-contamination occurs when unwanted material is transferred between two or more physical evidence sources. The third issue and challenges is unreliable techniques. There are a few disadvantages of using autosomal STR analysis for DNA profiling for rape cases, which is start with, after 3 days of the postcoital, the presence of a scarce amount of male DNA means a huge amount of female DNA. Thus, low sperm count, low mobility of sperm, degradation of sample, viability of sperm cells due to their fragility, sperm loss during DNA extraction, presence of more than one semen donor, presence of other bodily fluids from both female and male such as saliva, vagina fluids, fingernail scrapings, blood in a semen sample, then sample that lack nucleated cells such as teeth, bones and hair shafts. The next issue and challenges is wrong handling of evidences. Inaccurate forensic evidence can have serious and unjust consequences for the suspect and even victims. It is the responsibility of law enforcement to keep important physical evidence safe, because wrong handling of evidences can cause the cases to be dismissed or thrown out. Last but not least, DNA evidence planting and fabrication. DNA evidence planting and fabrication is one of the serious issues that occur as it is challenging to expose. Although cases have been recorded where law enforcement authorities have planted and fabricated evidence, there are also recorded cases where criminals fabricate DNA evidence to prove themselves innocent or to frame for someone else. One of the cases involving DNA evidence fabrication is the case of Dr. Sean Schneeberger, who was accused of drugging and raping his patient, Candice Fornagi. However, the semen found on the victim did not match Dr. John's blood, which was taken from his arm, causing the case to come to a halt. He was then convicted later on for this crime when he was accused of drugging and raping his 15-year-old daughter. This time, his hair and blood from his finger were tested and it matched not only semen from the current victim, but also with the Candice Fornagi, the first victim. He admitted later that he had been storing another person's blood by implanting a 15cm Penrose strain into his arm together with anticoagulants to prevent degradation. DNA theft can be done easily, and the acquired DNA evidence can be planted in a crime scene or on a victim to falsely link an individual to a crime. Only a small amount of DNA from an individual is needed to plant and frame them onto a crime. Next part is recommendations. The first one, recommendations for insufficient data for DNA profiling in Malaysia. There are 4. First, include more individuals, which is the number of hits goes up when the number of DNA profiles stored is higher. Second, include more convicts, which is only around 30% of convicts contribute to one of the highest numbers of hits. Third, introduce another genetic marker such as YSTR and mitochondrial DNA analysis. Fourth, funding, which is DNA profiling takes time, labor and cost. As for contamination, preserve the crime scene as soon as possible, wear proper clothing like PPE, and never leave anyone alone at the crime scene. Meanwhile, in laboratory contamination, the first recommendation that can be done is having set up PCR file from PCR product area. Second, keep PCR products and PCR regions separately. Third, do not even flick the PCR tube. And fourth, keep attire and work area clean always. Next, the recommendations for unreliable techniques. For example, the first one, YSCR analysis. Its benefits is to identify the male DNA by providing knowledge of the lineage of male relatives. It also cannot be used as a tool of identification alone. And complex mixture containing multiple semen donors, samples of male DNA in a mixture of bodily fluids, and samples such as semen from an azoospermic male and non-sperm DNA in low concentrations. It also recovered DNA profiles from prolonged interval post-coitus samples, sperm samples that have been degraded, and samples containing very little male DNA and missed female DNA. Next recommendation for unreliable techniques, which is second, mtDNA analysis. The first one, analyze degraded or low amounts of DNA samples and biological evidences which lacks nucleated cells such as bones, teeth, hair shafts, blood, and semen. As for wrong handling of evidence, place the evidence in paper bags or boxes. Put clean paper on every fold. Every dried stain should be kept in different envelope or paper bags. All evidences should be packed separately even though it is the same type. And victims should not remove evidences before examination. Seal and label accordingly. The last recommendations, which is for DNA planting and fabrication. The first one, collect additional evidence. Second, collect reference samples, such as collect from 2 different sources. Third, presence of authentication assay, which is differentiate artificial and natural DNA as natural DNA. Last but not least, DNA theft should be legislated as a crime to prevent DNA evidence fabrication from being overlooked. Conclusion: DNA's profiling's ability to identify the perpetrator in rape cases based on biological evidence such as saliva, blood, semen, epithelial cells has made it an important tool which however should always be evolving to better serve the community. Eliminating human error, using complementary analysis, and better equipping our national DNA data bank can be the start to having a more established DNA profiling system in Malaysia. That's all. Thank you.